unit basically. So the first chapter we are doing is chapter 5. Okay.
Yes.
Yeah. And in the last class, I think Rabia was there in the last class, right? Rabia?
Yes, sir.
Yeah, perfect. So would you like to give a quick recap? Let me just orient Aisha to this chapter, and then you just give a quick recap of these 4 terms that we discussed towards the end of the last class. So Aisha, we finished chapter 4 in the last class, okay? And we decided to go to the genetics unit first, and then we'll come back to the plant reproduction part, which is chapter 2. So we have done 1, 3, and 4. We'll come back to 2 later. We'll first do the genetics unit because this is a very heavy unit in terms of concepts and also the distribution of marks. Okay. Is that fine with you, Aisha?
Yes, sir.
Perfect. So yes, Rabia, would you like to give a quick summary of what we discussed about DNA, RNA, genes, and chromosomes in general in the last class? Just this much.
So actually, in the last class, I was out. So I was attending the class from outside, out of things.
Uh-huh.
So I actually did not write what all was given. I'm so sorry.
Okay, no worries. But did you understood the basic concept? It's not about writing. So let's me and you discuss so that Aisha can get a sense of what we did in the last class. Okay. Yeah, Rabia, how does that sound?
Sure, sir.
Yeah, cool. So I think in the last class, I told you that before we go and discuss in deep the genetics and what genetic mean, what are the discoveries, experiments, scientists, their contributions, it's very important to understand the concept of DNA and RNA first. Yeah. So let's go on with DNA and RNA first. So we discussed something about what is DNA, where is DNA in the cell, what is RNA, where does that come from, right, Rabia? So what is DNA and where is it in the cell? Is it present in some special cells or is it present in all the cells?
It's present in the nucleus, right?
It is present in the nucleus, right? Nucleus of all the cells, right? Is there any cell type in human body that does not have nucleus or its DNA? Rabia? Iram has also joined us. Good evening, Iram. How are you?
Good evening, sir. I'm good, sir.
Great. So we are just discussing what we summarized in the last class about DNA, RNA, genes, and chromosomes. So you can also help us because Aisha was not there in the last class, so we are orienting her to these concepts. So my question is, Rabia said that DNA is present in the nucleus of every cell in our body. So is there any cell type in human body that does not have nucleus or that does not have DNA? Or do all cells have nucleus and DNA?
RBC does not have any nucleus.
RBCs, you mean red blood cells, do not have a nucleus. So how do they form then? Because RBC is a cell, it must have come from some basic code, right? Some information. So how does it form without a DNA and a nucleus? How will a cell, whatever proteins are there in that cell? So RBC has proteins like hemoglobin, right? A very crucial protein called hemoglobin is present on the surface of RBCs, right? And we know that RBCs make that protein, put it on their surface. So how do they make their protein? How do they make that protein if they have no DNA and no nucleus? So yes, Rabia, Iram. Aisha, do you think that what Iram is saying is correct? Do you believe that RBCs do not have a nucleus and a DNA?
Yes, sir.
Yeah, so how do they make their proteins? RBC has a lot of proteins inside it. How does it make its protein? What could be the logical explanation? So you all agree that RBCs do not have a nucleus, but you also all agree that RBCs have a lot of proteins inside their cell. We know from our basic fundamental knowledge of biology that RNA comes from DNA and protein comes from RNA. So how does it all fits together? There can only be one logical explanation, and that is what happens. So what do you think? What do you think, Aisha?
No idea.
No idea. So what happens is there's only one way it can work out, right? It is not possible for any cell to begin without a nucleus, right? Then it's not a cell. So RBCs also, when they are formed, when they are prothrombocytes, like before, when they are, what are RBCs called? They're called yes. So when they are pro-erythrocytes, like before they come into circulation, where are RBCs formed? Anyone quickly? Human physiology.
Bone marrow.
Bone marrow.
Yes, bone marrow, right. So they are formed in bone marrow and then they are sent in the circulation. What is the average lifespan of a RBC?
120 days.
120 days. But out of that 120 days, it stays in the circulation only for around 45 to 50 days, which means more than half of its time, it's not in circulation. Okay, so it's not serving the purpose for which it is being developed. It is in the bone marrow, it is being developed. For that, in that time, it makes a lot of proteins inside it, stores a lot of protein, every protein that is required. At that time, when it is in the bone marrow, it has a nucleus. But when it becomes mature and when it is ready to come into circulation, the nucleus degrades. Okay, so the nucleus is not present anymore. So when it comes in the circulation, it is devoid of any nucleus, but it has all the requirements full and ready to survive for the next 45 days. And that's why RBCs have a fixed life span. That's why they die after one point of time, because they cannot make new proteins. So they only survive off what they made when they were in the bone marrow. Does that make sense, Aisha?
Yes.
Yeah. So it had nucleus when it was developing. A mature RBC does not have a nucleus. Okay. And then it serves for a limited time period and then dies. That's why it's a very good practice to keep donating blood, because anyways, you will be forming new RBCs every now and then. So whatever RBCs you have, if you donate them, your body will form new, and it can help people who need blood in cases of accidents or in conditions where they cannot form their own blood, or in surgeries, etc., etc. So donating blood is a very good practice. Okay, cool. So coming back to DNA, right? We have also Mariam joining in. Hello, Mariam, how are you? Okay, great. So I'll just check. Yes, so the spotlight feature is on for everyone. Perfect. And then RNA comes from DNA. As we know, DNA is double helix, RNA is single helix. And genes and chromosomes we discuss. These are the most important things we are going to talk about in this chapter a lot. So genes are nothing but segments of DNA itself. But any segment of DNA cannot be called a gene. There has to be some defined criteria to call a segment a gene. Not all segments are gene. We will talk about it today. And then chromosomes are supercoiled form of long DNA molecule. So DNA is very long a molecule, and it's a polymer. That's why it's very long. and it needs to be stored in a very tiny space inside the nucleus. So along with some proteins, which are called DNA binding histone proteins, this super long DNA is made super short and is coiled in the form of a chromosome. It's called super condensed form of DNA. And that's how 46 DNA pieces can be accommodated in each and every cell of human. Okay. So chromosomes are DNA threads bound to protein molecules to be condensed. Okay. All of these are clear to you?
Yes, sir.
Perfect. Now let's start with today's class. And in today's class, I will start with some fundamentals of sexual reproduction and variation, right? So the last top unit was all about sexual reproduction majorly. We have studied sexual reproduction in human in detail. And people knew from a very long time, like very, very early on, when science was just in the cradle and could not, was not up and running, people believed in philosophy, thinking, free thinking is called philosophy, where you try to find answers for phenomena that is happening from very early times, even thousands of years before Christ, before the modern calendar. People knew that the reason for variation, which means so many different forms of life of any given organism, human, dogs, cats, horses, everything around us, when they are so varied, that reason lies somewhere in sexual reproduction. Now, what they did not know is how it works. What is the mechanism? But they had hints that organisms that reproduce sexually produce the most variations, whereas organisms that do not reproduce sexually, or if you propagate organisms without sexual reproduction. And what are the ways to propagate an organism without sexual reproduction? Just name quickly a few. Just quickly name a few methods to propagate an organism without sexual reproduction. People, you have studied all that in class 11th, I'm sure, or 10th, like some basics. We can propagate plants through asexual reproduction a lot. We often do that. Cutting, layering, grafting, right? All those are artificial methods of, right? You have studied all this?
Yes, sir.
So what is that method called? Vegetative propagation, right? Just cut a stem of a plant and it will propagate, or you cut a ginger into a piece and put it in soil, it will grow from its own nodule. You can cut potatoes into two pieces, put it in soil, and it grows back from the same potato. Yes or no? Onions can also be propagated through that. So that's not a sexual mode of reproduction. That's a propagation through asexual modes. And there you don't see much variation in plants generation after generation. Okay. So having known that sexual reproduction is the key to variation, what people next try to figure out is how it works. Okay. Now the reason, as of now we know, is the DNA that is carried by gametes, and then meiosis, which is a special kind of cell division. You know about meiosis, right? We studied meiosis in the last class. Yeah. So meiosis is the reason for variation. There is a term which comes in meiosis. Can anyone tell me what in meiosis leads to variation? It's called
Crossing over.
Yes, perfect. Recombination due to crossing over of what?
The two non-sister chromatids.
Yes, non-sister chromatids. Now, when I say these terms, if anyone feels like that they don't know any of the terms on, or they're not able to understand, just please stop me right there and ask. it's your right, okay? Don't think that you will be judged, because even if you ask something 200 times to me, we'll just discuss it every time. But you should ask, because that's why you are here in the class. These classes should not be an obligation for you that you have to just join and sit, ask, okay? And on top of it, always remember it's a service you are availing. I'm teaching you because you are availing my service to teach you. So maximize the utilization. Everyone understands crossing over, non-sister chromatids, these terms, if not the process, but everyone is familiar with the terms?
Yes, sir.
Perfect. So this event, which happens during...
Mitosis and meiosis.
Sorry. Yes, Mariam, come again.
Also, can you explain the difference between mitosis and meiosis?
Mitosis and meiosis. Yes, perfect. We will go through the difference between so mitosis, in short, versus, let me write it first. Meiosis. So mitosis is also known as equational division, okay? And meiosis is called reductional division. Now I'll give you an example to explain what happens. Everywhere in your body, all types of cell divisions that happen for growth, development, repair, or any function, making blood, immune cells, proliferation, healing from a wound, all these cell divisions happen through mitosis. So all these divisions are mitotic divisions, where one cell, so this is one cell, it has the ability to divide, and then it will divide from the half, and it will form two daughter cells. And this cell will be diploid 2n. So the two daughter cells also will be diploid each, 2n and 2n. So from the mother cell, the ploidy remains the same. That's why it's called equational division. Okay, this is how all the body function, like all normal divisions in our body takes place. Is it clear?
Yes.
Meiosis is reductional division, and it's a special kind of division that only happens during gamete formation, okay, in sexual reproduction. In this, a mother cell, let's say it is diploid to begin with, finally, after the end of meiosis, so meiosis itself has two cycles in it, meiosis 1 and meiosis 2. And that's how one meiotic cycle completes. And by the end of one full meiosis cycle containing two subcycles, you will find 4 daughter cells, not 2. Okay. Is it clear?
Yes.
And each will be haploid. So this is reductional. It started with a diploid mother cell and it got reduced by half. Make sense? Why it's called reductional division?
Yes.
So mitosis, you can write, it happens for growth, development, proliferation of immune cells, formation of blood cells, everything. Maintenance, wear and tear maintenance. Everything happens through mitosis. Okay, is that okay, everyone?
Yes. Thanks.
Yes, and meiosis, you can write, it's a special type of division that only happens during gamete formation in sexual reproduction. Okay. Is that okay, Mariam?
Yes, sir.
Perfect. Okay, so where were we? Yes. So we know that in sexual reproduction, this is the reason, and we'll talk about it in detail. Now, before I go forward, let me spend some time on genes. So as we discussed, genes are segments of DNA, right? This is what we came up with in the last class. Now, can any segment of a DNA, if I take a long DNA and I cut from here to here, Will I call this segment a gene? For that matter, I take a long DNA, I cut it anywhere. Will every segment be called a gene? What are genes basically? What do they do? Any idea, people? Aisha?
They carry information.
They carry information. Yes, perfect. They carry information for what? You're right.
For any character like eye color, skin color, various things.
Yes, perfect. So a gene carries information for a particular character. Now, these genes are present, if they're segment of DNA, they get transcribed into RNA, right? And then from that piece of RNA that is transcribed from a gene in the DNA, a protein is formed, and that protein functions to give that character. Okay. Like she said, the color of eye will be because of a pigment. And do you know that how many types of eye colors are there in humans? How many eye colors are there in humans? Approximately, you don't have to be exact. How many can you think of?
4 to 5.
4 to 5, right? Yeah, perfect. So how many different kind of pigments should be produced to give all those eye colors? What do you think? If there are 4 to 5 eye colors, let's assume, then how many pigment genes should be there? How many different types of pigment should be there to give rise to those 4 or 5 colors? That's not a difficult question. If you have to paint a zebra crossing, how many colors will you take with you?
2.
So if there are 4 to 5 eye colors, how many pigments will be required to give those eye colors?
4 to 5.
4 to 5. But that's not the case. Okay. That's what intuitively everyone will think, because when we look at eye color, we think it's green, gray, brown, black, blue, right? Actually, these are all gradients of the same pigment. The same pigment, if present in very low amount, can give an eye a slightly grayish, bluish, and greenish texture. Okay. And the same pigment, if present in abundance, can give a eye color brown, dark brown. And actually, if you see under a, that's one exercise you can do. How many of you believe your eyes are black? Your eyes are black colored. Anyone's eyes black colored, according to them?
Yes, sir.
Yes, your eyes are black colored, you know?
Yeah.
Yeah, no eye is black colored. So all you have to do is take a flashlight of your mobile phone, open your eye, and just put some light from the side, not exactly from front, from the side, and look into the mirror. You will realize what you were thinking as black is actually a very dark shade of brown. Okay. Your hair can be black, but not your eye color, mostly. Okay, will you do that?
Why is like,
Sorry, anyone? What's it?
why the eye color cannot be black?
Because it's not a black pigment. That's why. In humans, right? The pigment is actually in abundance. It gives a dark brown shade. It's so brown that light doesn't get reflected back from it, so it absorbs the light. You know that why is eye designed like this? So eye is like this because this iris has color. In the center there is a hole, right? There is a lens. Through this hole, light goes inside, but everywhere else there are pigment that absorb the light mostly. Okay, if these pigments are very less, some light reflect back. That gives us a kind of illusion of a blue or a gray, greenish or a grayish eye. But if it is present in some abundance, it gives brown texture. If it is too much, it gives black texture. But basically, it's not black. Okay. So, but similarly, the gene which decides this eye color is the gene for this pigment. Now this gene must be present somewhere in the chromosome, let's say. And this gene should have a starting point, right? Otherwise, how will the machinery inside the cell, the proteins will know where to start from and where to end, right? So it should have a start and it should have an end. And if you make a RNA from this segment of the DNA, so this RNA will carry a message, so-called messenger RNA, and this will become a protein, which will be for the eye pigment. And every time the same piece of DNA will be transcribed to make the same RNA, and the same RNA will make the same protein. That's how you maintain continuity and exactly the same protein being formed all the time from birth till death. Yes or no?
Yes.
Yes. So clearly, gene is not any segment of DNA. Gene is a very defined segment of DNA with a start and an end. And this start of the gene, before the start of every gene, there is a region which codes for this sequence AUG in most of the genes. Okay, almost all. Like at this level, you just understand AUG, and you have studied about AUG before. Do you know about codons? No, right?
No.
No,
Okay. So AUG, just remember, this codes for, you know about amino acids, everyone?
Yes, sir.
Yes. So this codes for an amino acid called methionine. AUG codes for methionine, and this is known as the start codon. This is the sequence with which every gene should start. And then it will go on to whatever sequence it is, G, G, T, C, A, T, whatever. At the end, it should end with either UAA, okay, or UAG or UGA. These three do not code for any amino acid. Okay, if they do not code for any amino acid, no amino acid can be added and no protein extension will stop there. And that's where a gene stops. And these are called stop codons. So there are 3 stop codons in human and 1 start codon in humans for every gene. Okay, make sense?
Oh, what's
Yes,
codon?
sir.
yes,
Sorry, come again.
What's codon?
Yes, I'll come to codon later on. Just so a triplet of these bases is called one codon. It's a code. Just for now, understand it's a code that starts and ends a gene. Okay, just like we start a letter. So how will you say that this is a letter written to someone and not just a message inscribed? Because it has a format, right? It starts with dear or respected something, and then ends with yours sincerely or thankfully. And just by looking at the top and the bottom, we can say, oh, this is a letter. We can also tell, oh, this is an informal letter, this is a formal letter, this is a business letter, this is a proposal letter, right? Just by looking at that formats. That's you have done in your literature classes, right?
Yes, sir.
Similarly, just by looking at these codons, the sequences, we can say that, oh, this is where the gene starts and this is where the gene ends. And everything in between is called the coding region that is being coded, that has the message. So that's a gene. So a gene with a start codon and codon, stop codon, and the coding region in between. So this whole setup is called ORF in biology. It stands for open reading frame. Very important aspect of genetics. So whenever we find, have you ever, so none of you got curious when they write in the books that humans have around 28,000 genes. You know, this organism has these many genes. Butterfly has more genes than human. It has around 33,000 genes, being a very small organism. So none of you thought that how did they figure out that it has so many genes? How did you count that humans have around 28,000 genes, right? It's a very valid question, right?
Yes, sir.
So they looked for these open reading frames in the whole genome. If these open, so if you find a sequence starting with AUG, so it will be continuous, okay? It will be like this. Let me give you an example. So I am here and U is uracil. So this is the sequence of a RNA we are talking about. Let me write a sequence of a DNA. Oh, sorry. DNA does not have U. Okay. Can you all see this sequence?
Yes, sir.
Now your exercise is, can any one of you tell me, does this contains a open reading frame or not? Does it have a ORF in it? Let's say this is a sequence on a chromosome. It's just running. You are analyzing this sequence. You are trying to find a potential gene. Is there a gene inside this sequence? Do you think there is an open reading frame inside this sequence? That's how we find genes in a sequence, human, any genome sequence. How many of you think that there is a gene inside it? There may also, it's possible that there may not be a gene. So tell me. Rabia thinks, what do Rabia thinks? Rabia, do you think there is a gene inside it or there is no gene?
There is a gene.
There is a gene? Okay, so without telling me, if you think that there is a gene inside, can you tell me, including everything, start codon, end codon, and the coding sequence, total, how many sequences are there in that gene? That will tell you if you have found it correct. It's very interesting that you can find a gene here, but okay. Anyone else think that there's a gene, an ORF here in this sequence? So remember, this is a DNA sequence. It might be your DNA, my DNA, anyone's DNA. Rabia thinks there is a gene in it. Aisha, Iram, Mariam.
So I think there are two.
You think there are two? Okay.
Yeah.
In this, very good, great. So you can tell me what is the length of both the ORFs? ORF1, ORF2. So if you think there are two genes, there should be two ORFs, right?
Yes, sir.
Yeah. So tell me the length of both. By the way, others, Aisha, do you understand what exercise we are doing, or is it difficult for you to follow? If it is difficult, I'll try to make it easier and we'll do it again. Aisha, Iram, Mariam, none of you said anything.
I think there's one gene, but I don't know about two.
Aisha think there's one gene, and what is the length of that ORF for that gene, including start and stop codon, if you have found one? So Rabia thinks there are two. Aisha thinks there is one. Iram and Mariam, what do you think?
So what if I found two stop codons? Will it be two genes or what?
That you have to tell me. If you found two stop codons, will there be two genes?
So this is confusing.
Of course, it's going to be. Oh, sorry, I made a mistake here. Actually, there can't be a U in the DNA, right? So there has to be T. DNA has thymine in place of uracil. Okay, so DNA has thymine and RNA has uracil. Okay, uracil and thymine both are very, very similar in their structure, except for just one thing, that there is a missing CH3, a methane group. Okay. This is the only difference. But they're very similar. Okay, anyways. Mariam and Iram has not spoken anything. Either they have not understood or...
But then in the ORS, there are U included, right?
Sorry, there are...
There is the letter U included, right? In the ORS.
Yes, you include. I'm asking you so that you count. So when you are counting the ORF, you include the triplet for the start codon and also the triplet for the stop codon, and tell me how many total base pairs, total bases are there in your gene that you think is a gene. No one? Iram, Mariam?
So,
Yes,
I don't think so. There's a gene inside.
You don't think there is any gene. So that is interesting. Rabia thinks there's 2. Aisha thinks there's 1. Iram thinks there's no gene. Mariam, what do you think?
I also didn't find any gene.
Sorry?
So I didn't find any gene. So I don't understand.
Your voice is not very clear to me, Mariam.
No.
So she's saying that she didn't find any gene.
Okay, great. So Iram and Mariam both thinks there is no gene. Rabia thinks there are 2 and Aisha thinks there's 1. Okay, Aisha, would you like to tell me your genes length? In terms of bases, how many bases your gene has? Okay, let's do this exercise. So there are 2 things that you have to understand. Okay. When you start reading, okay, from here, you can find one code which codes for methionine. So when you have to find a code, you have to convert this whole thing into a mRNA, because mRNA is what gets transcribed. So do you see this ATG here? When it will form an mRNA, it will become AUG. So we got our start codon, right?
Yes.
But what you have to keep in mind is that beyond this point, once you have decided and defined one codon, everything will go in 3. So this, sorry, I lost electricity. It will be back, but I think you can hear me, right? My internet is still on.
Yes.
So ATG then becomes TTA. And when TTA will be, what do you say, transcribed, it will become UUA. Is UUA a stop codon?
No.
Right? No. So we are good to go. Then AGG, CCA, AAT, GAA, CAT, and then TG. There will be something which is not complete. But do you find any stop codon here? We did find a start codon, so it's very good that gene can start from here. But do we find any stop codon?
No.
No, right? So in this particular sequence from here till here, there is a start codon, but there is no stop codon per se. Okay. So there is no G, no ORF, because the open reading frame starts and ends with a start codon and a stop codon, and it maintains a codon basis like a sequence. Okay. Now what you see here, let's forget about for a second about the start codon. And some of you got confused. Why I gave this sequence was exactly to confuse you, because you learn a lot through confusions. Some of you must have figured out this TGA sequence, right? When this
Yes,
will
sir.
transcribe, it will become UGA, right?
Yes,
Is
sir.
that correct, Rabia? Aisha? Yes. So UGA is a stop codon, right? So we are good to end our gene here. Very good. We found a stop codon. But now, because you have found a codon, you have decided that this 3, then this 3 will be read. This 3 will be read. You keep going back, but do you find any start codon then? Because now you can't read ATG together. You have to read this GTT and this GAT, this TAC, and then these 2 will form something else. But do we find any start codon here?
No, sir.
So again, even through that kind of a reading, we do anything, we do not find a ORF. Okay. Now there is one way that there could have been a gene. Now, very interestingly, I'll tell you using this example only, how sometimes new genes or new ORFs are created. Now, let's say because of a mutation, there is an insertional mutation. Something gets inserted between this G and this G, and it is a A. Now, if this happens, and we start from reading this frame, so this is AUG. Now we'll read TTA. Now we'll read AG and this A, right? AGA, GCC, AAA, TGA, which will become UGA, right? So just by a insertional mutation, one extra base pair got added, right? And because of this extra base pair getting added, we have both a start codon and a stop codon in place. Do you understand? And also the triplet code is maintained. Now the region from here to here becomes a ORF now, and hence it can be transcribed as a gene. Make sense? Now this is a very small example I gave you. It will be thousands of base pairs. Genes are very, very long. But does it make sense? Do you understand something? Aisha, Rabia, Iram, and Mariam.
Yes, sir.
Yes, sir.
Yes, no?
Yes, sir.
Yes.
Yeah. Mariam also yes?
Yes, sir.
Perfect. So that's how genes are found out. So you can define genes, write down. It is a segment of DNA containing an open reading frame. It is a segment of DNA containing an open reading frame that can be transcribed and translated. That can be transcribed and translated into a functional protein. That can be transcribed and translated into a functional protein.
Can you repeat?
Yes, that repeating part is difficult, but let me try. What did I say? It is a segment of DNA containing an open reading frame that can be transcribed and translated into a functional protein. Okay. Is that clear, everyone?
Yes, sir.
Perfect. So gene is clear. Now let's come to inheritance and start the chapter finally. Everyone on the same page with bases and fundamentals?
Yes, sir.
Now let's talk about this person, Mendel. And his full name, complete name was Gregor Johann Mendel. How many of you know about Mendel? Who was Mendel? Gregor Johann Mendel. Who was he?
Father of biology.
Father of biology? No, he's not father of biology.
Genetics.
Yes. Biology started much earlier. He came up with something so radical that in the first place, people who were doing biology during his time did not accept what he did. They thought, okay, he did something and he does not understand biology. So actually they could not understand what he did and could not, accept it as biology. So he's called father of genetics, the modern genetics that we use. Okay. Now, Mendel was a physics student. Okay, he was a physicist by training. His education was in physics. He liked mathematics a lot. His liking was in the field of mathematics, numbers. By profession, he was a priest in a church, and he did his research in biology. What a diverse CV, right? The best CV one could have, right? So he had knowledge of physics, he had knowledge of mathematics, he was by profession a priest, and he did genetics in biology. And he's called father of genetics, of course. So he was the first of the people who actually used the concept of mathematics to explain biology. Okay, he used the concepts of mathematics to explain biology. In the initial days of science, science was mostly exploratory and observational. Okay, people did not used to do hardcore, experiments in biology. They just used to record, report, and talk about various different kinds of life forms, their shapes, their color, their changes, life cycles of insects, birds. Basically, in the initial years, biology was all about classifying and, classification and nomenclature, naming things in biology, putting them in order, putting them in categories, which animal should come together, which plant should come together, like that. So no one thought of using mathematics to explain phenomena in biology. That's what Mendel did. So Mendel worked with, what was the model organism that Mendel worked with? Yeah, so he used the model system called pea plant. Anyone knows the scientific name of pea? It's also
Pisum sativa.
Yes, it's a good thing that you start talking about scientific names. It helps you at the end of the session. It's called Pisum sativa. This is what he worked on. Same piece that comes probably to your homes. Now, first question that comes in board, a very popular question. Why did Mendel choose Pisum sativa? Let's keep solving some questions on the way. Why Pisum sativa? Anyone knows the answer?
It has a lot of variation.
It has a lot of variation. All organisms have a lot of variation. He could have taken a more varied plant. No, when you look at a pea plant, anyone has looked at a pea plant? Anyone has seen a pea plant?
Yes, sir.
In reality?
Yes, sir.
Yes. So when you looked at a pea plant, did you think, oh, this is the most variety plant in the world, so let me use this? Is there anything special about pea plant in the first look? When I saw the pea plant, it looked pretty normal to me, like any plant, any green creeper and climber with green stem and green leaves. Even the money plant looks the same, kind of. So of course, it has variation because it's a sexually reproducing plant, but all other plants have variations. Mango has so much of variation. Why didn't you work on mangoes? But you're right, it has variation. So let me keep that point, and what you're trying to say, let me make it a neater point. By variation, you mean it has
Different
variety of, yes, variety of traits, right? Variety of traits that can be studied. Correct? You're right. But that's it. A lot of plants have variety of traits. When you do science, will you just go for variety of traits?
It was easy to grow pea plant.
Easy to grow. What do you mean by that? Easy to grow.
Easy to cultivate.
Yes, same thing. But what do you mean by that? Again, there are many plants that are
Like we could perform experiments in a short period of time.
so short period of time is a third point. You mean it has a short life cycle, right? Short life cycle. So it gives rise to progenies very fast. That's true. It does not have the shortest life cycle, but okay, comparatively shorter than a mango or a neem or any other plant that could have chosen. But okay, what do you mean by easy to grow? Someone said easy to experiment.
Cultivate.
Yeah, so ease of sexual manipulation. What do we mean by that is you can easily, because pea has bisexual flowers, and you can easily do some procedures called bagging and emasculation. So do you know about these two things?
Yes, sir.
Yes, sir.
Who knows? Tell me, what is emasculation and what is
It
bagging?
was the covering of the plant so that it does not get infected.
Infected. I'm not sure if.
Um,
What are you talking about? Emasculation or bagging? Aisha. Yes, I'll come back to you, Hiram. Yes, Aisha. You're talking about emasculation or bagging?
Bagging.
Bagging. So you think bagging is for prevented from infection? Then how does it help in sexual manipulation or easy to grow, easy to experiment plant? Yes, Hiram. Back to you. Are you there, Hiram?
yes, sir. So emasculation is the removal of anther,
Okay.
the male part. Basically, pea plant is bisexual, so you do emasculation to remove the male reproductive part, and bagging is just to cover the flower so it doesn't get pollinated by other plants.
Yes. So in a way, what she's saying, do you understand what Hiram is saying is, if you're doing emasculation, you take a pea flower. So this is a pea flower. It has a stigma and it has anthers, right? So you cut down the anthers. So now it only has stigma, the female structure. That's emasculation. Make sense? So write down. Emasculation is cutting down of anthers in a bisexual flower. In a bisexual flower to prevent self-pollination, correct? Yes or no? Emasculation prevents self-pollination. So you cut the anther. So the same anther cannot now fertilize the same stigma on the flower. So it prevents self-pollination, but it promotes what?
Cross-pollination.
Cross-pollination. Self-pollination is called?
Pollination between the same plant.
Yes, there's a term for it.
Autogamy.
Autogamy. And cross is called?
Glutinogamy.
Xenogamy. Okay. Geitonogamy is? What is geitonogamy? Anyone knows geitonogamy? What is geitonogamy?
The same plant.
Sorry?
Different flowers of the same plant.
Yes, different flowers, but the same plant. So basically, it is within the plant, but between flowers. So it's not pure cross, right? Cross is xenogamy between two different plants. So it prevents self-pollination, promotes cross-pollination. Bagging, on the other hand, is where you just put a bag around the flower so that no pollen from outside can land on the stigma. So here, what are you preventing? Prevents cross-pollination. And what does it promotes? Self-pollination. Is it clear? So through these two processes, emasculation and bagging, which can easily be done in a pea plant, it becomes a very good model system to study genetics. And that's why Mendel chose it. First, it has variety of traits to study. And Mendel, for his study, chose 7 traits that he studied. And it has short life cycle, and it produces at the end of that life cycle a large number of offsprings. Seeds. Plants' offsprings are seeds, right? Because that is where their embryos are. That seed germinates and becomes a new plant. So it has a large number of seeds or offspring. So for any genetic study, you would like large number of progenies in the next generation from which you can derive a meaning, right? Yes or no?
Yes, sir.
Yeah. Cool. Anything else about pea plant that is special? And all these traits, one other thing that is special is it has variety of traits, and all the traits were contrasting. All traits have contrasting features, which makes it easy. For example, let's talk about, so these are the 5 reasons because of which he chose Pisum sativum. And what are the traits that he chose? Anyone knows the traits? So first trait was height of the plant. And the height had 2 choices, 2 contrasting features. Either the plant will be tall, which we represent by capital T, or the plant will be dwarf, that are represented with small t. Now, this is a wrong nomenclature given in your NCERT. Mostly everywhere they use this. But let me tell you the correct nomenclature, which is not in NCERT. Genes, when you go for higher studies in genetics or you want to become a geneticist, genes are always named after the recessive character. Okay, I have not told you about dominance or recessive yet, but this tallness is dominant. Okay, here in NCERT they use capital T, but actually it should be capital D for tall and small d for dwarf, because it's named after the recessive character. But because it will be very confusing at this stage for you, so they kept it like this. So you also have to follow this for your boards, okay? Just to know if you, in case, are interested. So first character was height, which had 2 contrasting traits, tall and dwarf. Second character was, tell me quickly. Sorry?
Color.
Color of what?
The seed.
Yes. Second was seed color. And again, there were 2 contrasting characters in that. Seed color was
Yellow and green.
yellow and green. Okay, in case of tall and dwarf, what is dominant and what is recessive? What is dominant will make an arrow up. So tall is dominant, dwarf is recessive. Okay. What does it simply means is, if any plant has this configuration of genes in the chromosome, capital T and small t, this plant will become tall. Though there is a recessive gene, there is an allele for dwarfness, it will not express itself in the presence of the other allele. So the allele which expresses itself successfully in the presence of the other allele. So this kind of condition is called heterozygous. Okay. Hetero means different. Heterogeneous mixture, homogeneous mixture, in chemistry you must have studied. So heterozygous means in the zygote, there are two different alleles, capital T and small t, on one chromosome, which come from father. Let's say there is capital T. On the other chromosome, which came from the mother, there is small t. But this capital T will express itself in the presence of small t. But this small t will not be able to express itself in the presence of capital T. That is the rule of dominance that we will talk about in some time. Okay. But one allele is clearly dominant, other is clearly recessive. Make sense, everyone?
Yes, sir.
Yeah. If anyone finds anything difficult, stop me right there and ask me to repeat. So seed color, in case of seed color, what do you think was dominant? So let's first what capital Y and small y. Okay. We will not write G. The same letter will be denoting both the alleles, one capital, one small. So if this is capital, it clearly tells that yellow is dominant and green is recessive. Capital also denotes dominancy in this format. Now the seed color of P, a dominant seed color is yellow. What is the color of seeds of the peas that come to your homes?
Green.
Green, right? If by chance you get yellow peas also come in the market. They're called something else. What are they called? Chickpeas or something. I'm not sure. Chickpeas are chole. I'm not sure. But they are used for entirely different purpose. But if at our homes, the green seeded peas come, you just clearly know that those are the recessive breeds. Yes or no? Yeah?
Yes.
Yeah. So those are recessive breeds just bred for market value and what people prefer to eat. Okay. What is the third trait that Mendel studied? Height, seed color,
Color of the flower.
flower color. Okay. What was the two different contrasting traits in flower color?
Violet and white.
Sorry, violet and white, right?
Yes, sir.
Or purple and white, you will just see. What is in your NCRT? Is it purple and white or violet and white?
Violet.
Okay. And which one is recessive and which one is dominant?
Violet is dominant.
Violet is dominant.
Capital V and small v. Right? This is dominant and this is recessive. Perfect. Next. Keep telling.
The shape of the seed.
Seed shape. Okay. I'm just arranging it in the order you are telling me. Okay. I'm not arranging all the seed together or something. Seed shape. What were the two seed shapes?
Round and wrinkled.
Round and wrinkled. A round seed. Which is dominant and which is recessive?
Round is dominant.
Capital R and small r, right? Dominant and recessive. Now you know that the peas that come at our homes are green but round. No one likes wrinkled pea seeds, right? So for one trait, we selected the recessive allele. For other trait, we selected the dominant allele and we made a hybrid. So all these peas that are grown in the market and which like companies like Shuffle send to your homes are all hybrid varieties made for what people prefer. Okay. Cool. 4. Fifth one is
White color.
Pod color.
Pod color. What is pod?
It is the cover in which seeds are
Yeah.
present.
So as a kid, we all must have gotten this duty of removing seeds from the pods, right? Pea pods. I used to get incentives. I used to do it for my mother. We all have done it at some point or the other, right? Anyone who has never,
Yes, sir. We still do.
we still do, right? I still do. When I go home sometimes, and if it's my mother's making something with peas, she's like, okay, come sit, and I think that's there. So this pod color again had two contrasting traits that Mendel knew. One was
Green and yellow.
green and yellow, correct? Which was dominant and which was recessive?
Green is dominant.
Green is dominant, so capital G and small g. Now you see here, in nature, seed are most dominant for yellow color, but pod is dominant for green color. So now at the end, we will see what kind of pea comes at our house. Mostly the peas that come at our house have green pod, green seed, and it is round, right?
Yes, sir.
Like everything green, it should be green. Perfect. What is the sixth trait that Mendel studied?
Pod shape,
Pod shape. Again, what are the shapes?
constricted and inflated.
Inflated and constricted. What is dominant and what is recessive?
Inflated is dominant.
Inflated is dominant. Okay. And constricted is recessive. Okay. 7. What is the last trait that Mendel studied? Sorry?
Position.
Could not catch. Sorry. No blue.
Flower
light.
position.
Flower position. Yes, perfect. Flower position. What were the two contrasting traits of this character?
Axial
Axial
and
and dominant.
Axial and terminal. Okay. Which was recessive, which is dominant?
Axial is dominant.
Axial is dominant, are you sure?
Yes.
Yes.
Terminal is recessive. Now, it simply means, what does flower position means? This is the plant growing. Either you have flower on axis, right here, left and right. This is axial position of flower. Okay. So this way, the plant can just keep growing because there'll be flower on both sides and it will keep growing. On the other hand, there is one trait where there are flowers at the terminal end. Also, there can be flowers here, not a problem. But then on the terminal end, terminal means at the end, there will be flowers. So it will just stop growing. Okay. Now, could you see that for all the 7 different traits he studied, it was very easily black or white, one or the other choice, which makes him, which made it very convenient for him to do genetics and do all the crosses and find some mathematical results and find out that how genes are associated with inheritance, and he became the father of genetics. Now, let me tell you a very interesting thing. When Mendel started doing his experiments, he knew nothing about genes. In fact, no one knew anything about genes. That no one knew that the DNA, people were trying to figure out what is the genetic material. They figured out what is genetic material. They also figured out later that DNA is genetic material. It transcribes into RNA and then translates into protein. At his time, there was no concept of gene known to people. So he knew that something gets transferred from one cell to another through gametes. And you know what did he called those things? Mendel used the term factors instead of genes. So he did not know that they are genes. He called that some factors. So in his personal research manual, where he has written and recorded everything, he's talked about some factors go from this kind of a gamete to that kind of a gamete and give rise to height, seed color, etc., etc. Okay, so he called them factors, very interestingly. Now let's talk about what he did. Everyone is clear till this point? Everyone on the same page? Yes.
Yeah.
Sir, how did he decide if the trait was dominant or recessive?
Yes, exactly. That's what we are going to do now. Now we are going to become Mendel, and we are going to decide whether a trait is dominant or recessive. Okay. It's a very good question. And that is $1 million question, that how will you say that? Because the definition says that if a pair of allele is present in a heterozygous condition, the one that expresses itself is the dominant one, and the one that cannot express itself in the presence of the other is the recessive ones. Okay, cool. But how will you experimentally find it out? So what Mendel did? Anyone has an idea what Mendel did? He did all sorts of process. He grew so much peas in his lifetime that he could have started a very, very successful company supplying peas across Europe. So what he did, he just observed in the beginning, and he found that one kind of pea plant grows very tall. Okay. Okay, keeps growing very tall. Whereas another kind of plant does not grow tall. It just grows this length and stops. Almost half the length it grows and stops. And he found that very, very particularly, in a very, very disciplined manner, either there were tall plants or there were dwarf plants. There was no plant which was intermediate, like this height, between longer than the dwarf and shorter than the tall. So there was no plant which was intermediate. So he found why no intermediate plant is there. This perplexed him. Now he decided to do one thing. He took a tall plant. Okay. Took a tall plant and crossed it with a dwarf plant. Okay. Make sense?
Yes.
So this is the parent generation. We call this F0. In genetics, the parent generation is called F0 because that is where we start our genetic study from. Now, just like any normal observer of science and biology of that time, you will expect, because when these two will be crossed, again, how can you make sure that they are crossing? You can just emasculate the anthers of one plant, let's say tall, and you can just remove all the stigma of the other plant that is, let's say, dwarf. So only cross-pollination will happen and no self-pollination, or you can allow self-pollination by bagging. So you can do all these sorts of things, and you can easily make a tall cross with a dwarf with surety, and all the progenies that will come. So in F1, what do you expect? Let's say if there are 100 seeds, which means 100. So he did a very, very tedious job. He crossed it, got all the seeds, collected all the seeds, then grew all the seeds. Okay. And then in the next generation, which is F1, he again saw that what is the ratio of tall and dwarf. What do you think normally should have happened if I cross a tall and a dwarf and there is no concept of intermediate in that species? Then you will say that some plants will be like, let's say this is the female and this is the male plant. When I say male and female, I mean that in this anthers were removed, in this stigma was removed, right? So when they are crossing together, you will say that some will be like this tall and some will be like this dwarf. Yes or no?
Yes, sir.
Yeah.
Yes, sir.
But instead, what happened, he was very surprised that all the plants in the F1 generation were tall. And now he was surprised that where did this parent's characteristic go? What happened to dwarfness? Why all the seeds are tall? Even the, like, all the, even, even in case, sorry, let me just erase this. This will make things more interesting for you. Even in case where the dwarf is the female organ containing and the tall is the male organ containing plant. Now this dwarf is going to produce seeds. Yes or no? Do you agree?
Yes.
You are saying no or you are saying yes? Just give me 1 second. If the dwarf plant has the female structure, this is the one which will have ovaries, and this is the plant which will produce now seeds. Yes or no?
Yes, sir.
But it has crossed with this plant. So even the seeds which come from the dwarf plant, when they were sowed in the soil, the F1, they all became tall plants. Now he was puzzled that from a dwarf mother, all the plants which are coming are tall. How is this happening and why is this happening? And does not matter if you keep dwarf as the female plant or tall as the female plant. Whenever you cross a tall with a dwarf, he found that in F1 all are tall. So that puzzled his mind and he realized that, okay, some factor is being transferred from this plant and some factor is being transferred from this plant, and whatever factor is coming from tall plant is a dominant factor. Only that matters. And whatever is coming from a dwarf plant is a recessive factor. He didn't know at that time the concept of genes or alleles. Does it answer your question, Iram? That how did he figure out
Yes, sir.
dominant and recessive? Yeah. Now, interestingly, what Mendel actually found out was this cross. When he took a tall plant, by chance, he took a pure line tall, capital T, capital T. So for this plant, all the alleles, both the alleles had a capital T, capital T. And he crossed it with a small t, which is a, we call it homozygous tall. And we call this a homozygous dwarf. Now I have a question for all of you. Can there be a heterozygous dwarf? Can there be a heterozygous dwarf pea plant?
No.
No, it's not possible, right? Why? Because the moment you make it heterozygous, only the dominant allele will express itself. Is that clear, everyone?
Yes, sir.
Perfect. So there can never be a heterozygous dwarf pea plant. So when you cross this, how many of you know about how to cross it? How many of you have learned about Punnett square in your school?
I have.
Perfect. Now, what you have to do first, I will go to Punnett square, but before that, what kinds of gamete will this homozygous tall plant make? So it will make one gamete. While making gametes, it will segregate the alleles, right? One will go, because gametes are haploid. So this will go into one gamete and this will go into another gamete. But both the gamete will be same only, right? Capital T, just like a human female, X and X. right?
Yes, sir.
So on one axis, we will write the male, and on one axis, we will write the females. So let's say this is from female. So a capital T and a capital T, two different kind of gametes will be formed. In case of this also, there will be two, like basically same type of gametes will be formed, capital small t and small t. And now when you, this is called a Punnett square. On one axis, you write one gametes from one parent. On other axis, you write gametes from other parent. And then you cross it, capital T, small t, capital T, small t, capital T, small t, and capital T, small t. So all four genotypes will be capital T, small t. So all four are heterozygous in nature. And can a heterozygous situation express the recessive allele?
No.
No. So the only possibility, if it's heterozygous, it will be tall. So all the plants genotypically were hybrid, but phenotypically, phenotype means how they look, all of they looked tall. That's how Mendel thought it can be explained, right? But he didn't stop here. Okay, what he did, he took F1 generation, the same siblings from F1, and crossed them together. Now this time when he did that, he realized now there are two different gametes, right? Capital T in one gamete, small t in another. Here also capital T in one gamete, small t in another. Now if you cross, you will find capital T, capital T, capital T, small t. Here also capital T, small t. But in this case, you will find a small t, small t. So you will now find genotypically, there are three different genotypes. Capital T is there, capital T, small t is there, and small t is there, correct? And the ratio is 1:2:1. This is the genotype ratio. But phenotypically, it does not matter if it is homozygous or heterozygous. If one dominant allele is present, these three will be tall and only one will be dwarf, right? So phenotypic ratio will be only 2. Tall is to dwarf. There is no intermediate in P. And what will be the ratio? 3:1, right? 3 tall and 1 dwarf. Is this clear, everyone?
Yes.
But pay attention. To make, this is called F2 generation. This is called F1 generation, and the parents were F0 generation. To make, in F1, he crossed a tall with a dwarf, and he got no dwarf, all tall. It was very interesting, right? He was very, very amazed. Okay, what is happening? Cool. Even more interesting is this. To make F2, when he self-crossed, this is called self-crossing of F1. Because, and in self-crossing, he used both tall, right? This is also a tall plant, and this is also a tall plant. When he used two tall plant, and when no one expected that there will be a short plant coming up, then he saw a dwarf plant. Isn't it interesting? And this observation was not possible to be explained without doing these mathematics and this symbolic genetics. So this is what Mendel achieved. He realized, so this trait of dwarfness, it skipped one generation. In F1, there is no dwarf. And then when you are not expecting it in F2, there is all of a sudden a dwarf. So that's how recessive genes were defined, recessive genes skip a generation. Okay. Make sense, everyone?
Yes.
So we'll stop here. I think we all now got a grip on what Mendel did. I'll start from this point, and tomorrow, day after tomorrow, next class will be a little more ratio heavy and genetics heavy. So please go and read about monohybrid and dihybrid cross from NCERT, and then we'll discuss on Wednesday. Okay.